Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Hello everyone, welcome to my YouTube channel. So today we are going to discuss about tissue engineering TRAID. So in the previous class we discussed about the introduction to the syllabus of the module 1, and we discussed about introduction to tissue engineering with two definitions and a few the explanation about tissue engineering along with its importance and the drawbacks of the conventional methods where they used to use like normal surgical reconstruction and organ transplant and all, and how exactly the tissue engineering can overcome all these things. So that's what it can be so we studied in the previous class. So in today's session we are talking about we will be discussing about tissue engineering TRAID. What do you mean by tissue engineering TRAID? So a tissue engineering TRAID is nothing but we are having three approaches. That's why we are calling it as a TRAID. So tissue engineering TRAID, what happens? So here first approach, second approach and third approach. We know that the three things are there, that is three things need to be used in tissue engineering. The first thing is the cells. That means cells, we need to have the cells so that we can grow them to form a tissue. The second thing is the growth factors. To grow the cells we need growth factors. Okay. And the third thing, scaffold or extracellular matrix, okay, which will hold these particular cells. That means you are having extracellular matrix, you take the cells, you seed in this particular extracellular matrix. Okay, this extracellular matrix itself can be called as scaffold. Okay, so this extracellular matrix will be like a kind of network structure. For example, this will give the mechanical support and structural support. For example, now the ear, we are having this shape, right, or nose shape. Every particular organ or any particular appearance what we outside, so it has got some particular shape. So that shape is maintained by this particular extracellular matrix or scaffold. So now in tissue engineering, in order to develop a new tissue, we need to take out that extracellular matrix first. You need to take out all the cells which is present inside that and empty that particular thing. Now the second thing is cells. Cells you need to implant or seed in the extracellular matrix. Now the third thing is growth factors, so that the cells will go and attach to the extracellular matrix or a scaffold, and growth factors, when you add, the cells will start growing faster. So these three things should be there. Cells should be there, growth factors should be there, the scaffold or extracellular matrix should be there, or biomaterials. Okay, scaffold, extracellular matrix or biomaterial, all three words will give you the same function. That means they will act as what? The natural extracellular matrix. That means they will give the structural and mechanical support. Okay, so let's start the topic. So I hope you understood the introduction. So biomaterials, so cells and as well as growth factors, these three things are the tissue, or known as tissue engineering triad. So tissue engineering triad, in a sense, we are having three important materials that should be there in order to go for tissue engineering, any techniques or principles. Okay, so the first thing is we are having biomaterials, cells and growth factors. These three things are there. The first approach, we can have the approach first, second and third, we can discuss anything. So first approach, we will take it as a cells, because cells are the basic requirement. In order, if there is no cells, if you are having only biomaterial or extracellular matrix or the growth factors, then what you will grow? So cells will act as an important aspect. So we will take it as a first approach. The first approach, cells can be used as a therapeutic agents, okay, to restore the functional tissue. That means whatever the functional tissue is there, to restore that we are using the cells as a therapeutic agent. Now this approach mainly involves the isolation of the cells from different cell sources, and also use stem cell therapy, okay, placing them in the site of the interest for improving the tissue function. So for example, now the cells need to be collected. From where you will collect the cells? So the maximum we have to think of taking the cells from the same person. If the patient is having some liver disorder, or if any liver is bit damaged, a part of the liver is damaged, then other part liver cells can be extracted and those cells only can be used. Understanding? So first you always go for the same person. The reason is very simple. When you are taking the cells from the same person or same patient and you clone it and you make a tissue, and then when you implant it back, so his body will not see those cells or a tissue or organ as a new foreign organ or any other organ which from the external. Understanding? When the body doesn't recognize, when the immune system doesn't recognize these cells as foreign cells or foreign tissues or foreign organs, then there is no immunological reactions or there is no immunorejections. So there are no any infections or inflammations which is caused by the immune system. Understanding? So immune system will see those cells or tissues or organs as our own cells, our own tissues or our own organ, because these particular cells are taken from the same person. If we are taking the cells, this happens in some cases where the cell, imagine the almost all liver is damaged. Second example, what happens? No cells are there or cells cannot be taken from the same person. Then they have to take from a different person. When it goes to the different person, then there will be a problem. Okay, because the different person cells, when you give it, when you use it, when you clone, when you make a tissues, okay, liver tissues or liver itself, and when you implant inside the human body will start, it's humans immune system will get activated. We will see that as a foreign object or foreign cells, so it will release antibodies, so antibodies will go and attack those cells. So in order to stop this action from the immune system, we will have to extra add the immunosuppressors. Understanding? So that's why first we have to plan for a particular same person only. So here this approach mainly involves in the isolation of the cells from the different cell sources, and also it can be, you can use stem cell therapy also, so placing them in a site of interest, improving the tissue function. So second approach is to the exogenous delivery of growth factors or growth promoting substances like growth factors. So how? By using carriers. So your carriers can be polymeric or lipidemic, whatever it is. So what these growth factors will do? They will stimulate. In name itself it is there. They will improve the growth. So of what? Cells. So they can stimulate the endogenous stem or progenitor cells for specific differentiation, thereby replacing the lost cells or tissues. That means they will stimulate, they will help the cells to grow. The third thing, that is biomaterials. Here the third approach is to use artificial three-dimensional scaffold, or you can say the extracellular matrices, or you can simply say matrices for the growth of the cells. The basic idea is to have a network or a structure which should give the mechanical and structural support to the cells. Cells, when you go, now imagine the half of the ear is cut. Now in order to gain this particular half ear, what I will do, I will take the extracellular matrix of this particular ear, so that I can remove all the cells from that. Now I will add the whatever the cells are here, I will extract from the same person, I will add here. Now growth factors I will add. Okay, now this particular tissue will start, cells will start differentiating 1 to 2, 2 to 4 like that, and then finally tissue proliferation moment will be there, differentiation, proliferation and development, and then growth, and finally you will get the whole bunch of tissue or half of the organ. Now this half of the organ or a tissue, now I will grow. It has to grow for a particular shape, right? This shape only it has to be done. So that is called as 3D scaffold or matrices. That means this particular, it will restrict the shape and size. It will decide, it will be the deciding factor for the particular shape and size. So that's what we will use for three-dimensional fields. So it can be extracellular matrix, it can be you can call it as 3D scaffolds or biomaterials, matrices, everything meaning is same. It should provide the structural and mechanical the support to the tissue growth. Next, limitations of these approaches. There are limited supply of autologous cells. That means we cannot have sometimes from the same person, if the cells, as I told you, if the whole organ is damaged, we cannot use those cells. And then the requirement of immunosuppressants for allogenic sources. That means if you are taking from the different person, you need to go for immunosuppressants, or else, because as I told you, when you implant from other person ka cells inside our body will definitely naturally it will produce antibodies. The immune system will produce the antibodies. These antibodies can come and harm those cells, kill those cells. So that's why we need to have immune suppressant, so that this particular immune system should be suppressed. So for the allogenic sources, so disease transmission associated with the xenogenic sources. That means if you are using for a different species, then the disease might be transferred from that species to this species. Then ethical consideration with the use of stem cells. So that is, these all things are the limitations. So what part of the body tissue engineering can replace? It can use for skin replacement, kidney replacement, vascular tissues, pancreas, thyroid glands, esophagus, adipose tissue, cartilage, liver, bone, nerve and bladder. All these tissues are as well as organs can be replaced with the help of tissue engineering. So challenges, what are the challenges? In conclusion, there are few challenges to be answered for the reconstruction of the functional tissues. There are 7 challenges being listed out. First challenge is, how do we develop a close approximate biological replica? That means if this is a particular organ, I need to have an organ exactly like this. So that is still a challenging task. Next, should the tissue be produced in vitro, implanted or in vivo? Okay, that means whatever the tissue has been produced in the laboratory, that means in vitro in the means laboratory, implanted in vivo, that means you have to produce in the laboratory, then you are going to implant in the human being. So that is, usually we will do these two techniques separately. Either we do it in lab, either we do it in vivo or in vitro. But here you need to grow here and then you have to implant here. That is also a challenging task. The third task is, what type of scaffold should we use? Okay, what kind of scaffold, what kind of biomaterial should be used? Next we are having, how do we manufacture? Okay, what particular technique or the mechanism we have, or the process of engineering can be used, okay, which will act as an optimum or which will give you the better structure. Then what cells are to be used, what kind of cells we are going to use? That is also very important. That is also a challenging task. So under what conditions can cells expand without affecting its phenotype? That is very important. It should not affect its phenotype, right? But it should still expand. So at what are the conditions need to be maintained in order to get this particular property? And then what regulators do we use to stimulate the cell proliferation and differentiation? Can we have a control over cell differentiation, or can we have a control over proliferations? If yes, then what kind of regulators we are going to use? These all are the several challenges still tissue engineering is facing, and it has to be addressed for the better applications of tissue engineering. So understanding for all these challenges, what should be there? Understanding should be there. So the solution is very simple. Hence, answer to all these questions or all challenges, it is necessary to understand the basic things such as how tissue gets organized inside the cells, okay, or inside the organ. That need to be understood. And what are the different basic types of the cells? We know that T cells, B cells, that, this and all. So, and different every organ has got its own different cells. Even those cells need to be understood. Then how do we classify the defects based on the tissue? So how we are going to classify? For example, skin, we are having the dermal, epidermal and transdermal. That is, similarly, if the dermal only gets destroyed, we will call it as a first case, or this thing, the small damage or something. Next, intermediate, and then we are having the third degree. So first degree, second degree and third degree. Third degree means almost the transdermal, it has reached all, it has ruptured all the cells, like that. So similarly, we need to have a different cells. So there are different cells in the dermis, when epidermis different cells, endodermis different cells, right? So that's why different cells need to be understood. What are the different cells which are involved, which are going to produce? Then how do you classify the defects? That means just now I told you about the example of skin, that how well can tissue repair its defects. That means the particular tissue should repair its repair itself, right? So how well it will repair. Then what are the components comprising the tissues? That means what are the things which are the particular tissue is made up of. All these things should be understood. Okay, so that was about the tissue traits and the challenges, and as well as the solutions for that, or understanding is required. So next topic we are having the current scope of development, that we will discuss in the next class.